Okay, hello Shaji. I heard you had a wonderful abstract presentation at the EHA meeting. I'm sorry I missed it. It was on the redirect study with Teclistamab and Talquetamab for patients with refractory myeloma and extramedullary disease. What did you find?
So, Vincent, thank you for having me. Clearly, patients with extramedullary disease are so hard to treat. We don't really have good treatment options. What we found in the RedirecTT-1 study, again, based on some early results, was again, combining the Teclistamab and Talquetamab is a very effective strategy with a response rate of 80% and a median progression-free survival of about 15 months. So, durable, deep responses in a hard-to-treat patient population.
Wow. And the Teclistamab and Talquetamab are given how often, and is it less than the dose we usually use for each drug?
So both are given in a every 2-week fashion. They're given together in one right after the other, after 3 step-up dosing. The dose of talquetamab is the same that we use in the every 2-week, which is the zero point 0.8 milligram per kilogram. And the dose of teclistamab is twice what we use in the weekly regimen, so 3 milligram per kilogram every other week. So basically, both of them every 2 weeks for 4 to 6 cycles, and then we go to once a month.
Got it. And how did the patients tolerate it? Like was there double the toxicities?
Yeah, the interesting thing with this study was that the toxicity profile was no different than what we would have anticipated with either one of these drugs. The hematological toxicities were common, but when you look at cytokine release syndrome and ICANS, they were mostly grade 1 or 2, mostly seen with the step-up dosing or with the first full dose. There are certain side effects that we attributed to talquetamab, like the skin toxicity and the altered taste. Again, similar frequency in the combination as we would have seen with talquetamab alone from the monumental trials. And when you look at the infection rate, in fact, the grade 3 for infection was about 22%, not much different, or maybe in fact a little bit less than what we have seen with the bispecific antibody used alone in some of these trials. So I would say toxicity profile very comparable.
Okay. And the efficacy, were you surprised to find that you've got a PFS of 15 months in this population? How does it compare to other trials with either each agent by themselves or other regimens?
Yeah, the efficacy was really, surprising when you think that the overall response rate with teclistamab or talquetamab used alone is about half of what we have seen
Okay.
with the combination. And more impressive was the fact that the complete response rate with the combination was 3 times what we would have seen with either tech or tal. And also put it into context, other therapies, we're talking about maybe a 3-month PFS with conventional agents, 6 months with some of the bispecifics, and 9 to 10 months with the CAR T. So we are talking about maybe, 15 months PFS. So again, with all the caveats of this being a single arm trial, the historical data would support this as something much better
Okay.
than what we have used before.
Why is it? Is it because like you are targeting to this, what is the reason why a bispecific against GPRC5D and another one against BCMA would combine and get you a synergy?
Yeah, I think it speaks to the biology of the disease. Extramedullary disease are genetically very complex and highly proliferative disease, just like what we see with plasma cell leukemias and so forth. So I think in order to get a good response, we have to go after two different targets. And I think the using both these drugs together allows us to kind of get a control over the entire clone, but giving them continuously allows us to keep that clone under control, unlike maybe some of the newer treatments like CAR T, where you do get deep responses, but there is nothing to keep the clone under check for a longer period of time, and they evolve fast and they relapse. Whereas using this approach, we are able to get a more durable control of the clone.
Okay. And final question for you. This is again very exciting. I mean, I'm really looking forward to seeing the full publication. I'm sure you're working on that. But where does this go? And also, why use Tec plus Tal compared to some of the trispecific, which seem to have target BCMA and GPRC5D at the same time?
It's a very good question. I mean, hopefully, this will get to a point where it has regulatory approval to use that. But again, I think this gives us a good sense of the biology we need to target, whether we target with Tec and Tal used together or a tri-specific, which clearly has very exciting activity in the relapse setting. It doesn't really matter. I think either one would be okay as long as we can encompass the biology with the therapy.
Got it. And you think that um, in a patient that we clinically have, that this therapy might become approved or available?
That is a hope that there's going to be a regulatory approval for use of these two drugs in combination for patients with extramedullary disease as defined in the trial, which is clearly an unmet need.
Again, I mean, extramedullary disease in my mind, particularly at relapse, when the relapse is occurring full blown with extramedullary disease, is quite difficult to treat, and it's almost like a secondary plasma cell leukemia, very very high risk myeloma. So I'm sure you're very happy to have a regimen that has such high activity.
Absolutely.
Yeah. Thank you so much, Shaji.
Thank you.